Alrighty, hopefully you are logged in and can see what I'm showing you. This webinar is to help you get ready for your upcoming test on tomorrow about DNA, RNA, and transcription, translation, replication, all that good stuff. Before I actually get into the webinar, I want to show you a couple of things on the online classroom so you know kind of what you need to be doing. First, if you have any questions during the webinar, there's two ways you can send them to me. You can, on the online classroom, fill out this questions box with your name, your question, and then click the submit button. And if you come up with another question, you can submit another question by going back to that if you need to. You can also send questions to me in an iMessage. That may get you a little bit of a quicker response by sending an iMessage to jasonmrpurser@icloud.com. Either way, that's fine. If at any point you feel that you are finished with a webinar, then you can close at any time. I'm not making you watch the entire thing. Just know that if you want to do the extra credit is going to be some questions that you would have to have watched the webinar to be able to answer, most of them. So if you are intending on getting the extra credit for this webinar, you will click on this link here, and it will take you to this page where you'll fill out the extra credit questions. And you can see what some of those are going to be. In the webinar, I'm going to show you a picture of a famous scientist. You'll need to put that person's name in this box. On one section where we do replication of DNA, there's going to be a repeating three-letter word, and it'll go in this box. I'm going to tell you on one slide what my two favorite colors are, and then there's some questions to answer about the actual content. Questions kind of like what you would see on your test tomorrow. Now, these first three questions are just kind of random things that will show me that you are paying attention. So if you answer those appropriately and you answer these questions appropriately, then you'll get up to 10 points on your test tomorrow. So that's how you do the extra credit. As far as the webinar, I'm going to go ahead and enlarge my screen where you can see a little better. We're going to be looking at a presentation, and as I start getting questions from you, then I'll pop out for a minute and I will answer those questions. If you come up with anything that you don't understand and you still don't get it after the webinar, then you can send me an email and I'll try to answer it through email, or you can come see me first thing in the morning. So in this review session, we're covering DNA, RNA, and protein with these topics in mind. We're going to be looking at DNA versus RNA and how they're different. We're going to be looking at replication, transcription, translation, and mutations. And I'm going to go in that order, and there'll be some questions for you in the extra credit that relate to some of these topics. If you have any questions about these, remember, you can either send me an iMessage with your question, or you can send me the question in the question box on the online classroom. So DNA versus RNA is our first topic, and this is one of the big topics on your EOC as well, looking at the differences and similarities in DNA and RNA. Both are nucleic acids. However, they do have various differences that make them physically and chemically different. DNA is a double strand, it's a double helix, whereas RNA is a single strand, just one helix, or one strand, one string. It's almost like a chain. DNA is long and kind of complex. It's big, so it can't leave the nucleus. But RNA is short and simpler than DNA, so it can leave the nucleus during translation or before translation. The sugar is also different, and the bases are different. For example, the sugar in DNA is deoxyribose. That's where the D comes from in that three-letter acronym. And the sugar in RNA is ribose. That's where the R comes from in RNA. So the bases in DNA are adenine, thymine, guanine, and cytosine. We just will abbreviate for the test tomorrow A, T, G, and C. And RNA contains adenine, uracil, guanine, and cytosine. A, U, G, and C. And again, as I said just a minute ago, DNA, because it's so big and complex, it can't leave the nucleus. So all processes involving DNA happen there. And all processes that involve RNA only, or RNA and protein, those can happen outside in the cytoplasm because RNA is simpler, it's shorter, and it can leave the nucleus. So for the test, you'll need to know these differences in DNA and RNA. About DNA structure, you need to know that it is a double helix, and the backbone of DNA, in other words, this part, the purple part on either side, is made up of sugar. That's what these little pentagons are. the sugar deoxyribose, and they're made out of phosphate groups. That's what these yellow P's are. So the backbone of DNA is sugar and phosphate, and the rungs of the ladder, so to speak, are the bases in the middle. So you can see guanine and cytosine, adenine and thymine. All of those are the bases that make up the rungs of the ladder. And one little piece, you see the orange box on the right-hand side, one little piece of DNA, one building block of DNA is called a nucleotide. One nucleotide consists of one base, one deoxyribose, and one phosphate. So nucleotides are the building blocks, or the monomers, in other words, of DNA, and they consist of those three parts. And all those different monomers come together to make up the complex molecule of DNA. Now, flashing in the left-hand corner of your screen is one of the questions for the extra credit. You need to be able to tell me what scientist I threw randomly into the presentation to show me that you did actually watch the webinar. Einstein has nothing to do with DNA, but that will be one of the extra credit questions. So the processes of DNA replication, transcription, and translation will all three be on your test tomorrow. And you need to know what happens in each one, where they happen, and what enzymes or molecules are working to make that happen. So we'll study replication. This is actually misspelled. There should be a P right there. Sorry. Replication is where the DNA gets copied and makes more DNA. The DNA is unzipped by an enzyme called helicase. And the helicase untwists and unzips that double-stranded ladder of DNA. Bases then get added to that open strand. And the way that happens is through the help of an enzyme called DNA polymerase. Helicase unzips, polymerase lays down the bases, and polymerase also proofreads that sequence to be sure there's no mistakes. And since this is happening with DNA being made into more DNA, we know that it has to happen in the nucleus because DNA is too large to leave the nucleus. I'm going to show you if this will work, an animation of this process. There we go. So this is my DNA template strand. And I don't know how well you can see this on your screen. If you're viewing the video and you go to full screen, it may show a little better. But you have the bases color-coded, and they also have different shapes showing that they fit together. And A's will only bind with T's, and G's will only bind with C's. So this DNA is about to be replicated or copied. And the events of that are the unzipping of the DNA. DNA gets unzipped on one end using that enzyme we called helicase. So that would be one of these two molecules here. DNA gets unzipped, and then the other enzymes start laying down bases on each side of the unzipped strand. So more bases are being added. Even more bases are being added. And we can see that that first half is already complete. All the A's were matched with U's, or with T's rather, and the G's were matched with C's. Notice there's some problems right here, the A's and the U's, because we're not talking about RNA yet. So this is a problem. Remember that polymerase looks for problems as it's proofreading, and it tries to cut those out and correct them. So we'll see if that happens. So there's the rest of my bases. And then polymerase is going to scan. Notice it fixed that problem. It's going to keep scanning, and it fixed a problem that was right here. And then it's just going to put all the pieces together, and now we have two strands of DNA that are identical to the original strand. So I'll watch that with you one more time just so we can see all the steps. DNA gets unzipped, bases get laid down. There are some mistakes made along the way, but polymerase comes and scans and fixes those mistakes. See the red, that indicates a mistake there. The mistakes are fixed, and we see the DNA gets put back together. So that's the process of replication. Replication is where the DNA is being copied into more DNA. I'm going to go back to full screen, and we'll move on. Replication, DNA making more DNA. It happens in the nucleus, and it uses helicase and polymerase. Remember, this guy down here in the corner is important. All right, so let's see if you can replicate this strand. ATC CGC, GTA, TAT. I'm going to go back out where I can actually type in this document now. And let's replicate this. A in DNA will bond with T, and T will bond with A. C will bond with G, G, C, G, C, A, T, A, T, A. Because in DNA replication, A's will bond with T's only. Now let's see if you can replicate this one at the bottom. This will be part of your extra credit. G will bond with C, T will bond with A will bond with T. Notice the pattern. GTA, 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 GTA. This little sequence is going to spell the word cat over and over and over again. That'll be important for your extra credit part. Can't spell. Your extra credit part of the webinar tonight. Remember the word cat. Hint, hint, hint. Okay. We'll go back to presentation mode and let's move on. Replication. DNA is being used to make another strand of DNA. Then we have transcription. Transcription is where we go from DNA to RNA. Both are nucleic acids, so we're speaking kind of the same language, except for that letter T gets substituted for a U. So in this case, DNA gets unwound and bases are added to the template strand, a lot like the previous example. But the difference is we're using RNA polymerase this time. Polymer means many units. So polymerase is the enzyme, ASE, enzyme, that is putting many units together. RNA polymerase is putting many units of RNA together. DNA polymerase from the previous topic is putting many units of DNA together. And all this is taking place in the nucleus. So again, transcription, DNA is converted to RNA. I'll show you an animation of that one in just a second. Let's see if we can transcribe this sequence. Remember, our base pairing rules changed just slightly. And A's will bond with U's in RNA. So here's our sequence. T will go to A, but A will go to U. And we have G, C, G, U, A, C, U, G, U, G, C, C, A. This is transcription. I've gone from my DNA sequence to messenger RNA. Remember, DNA is too big to leave the nucleus, but RNA can. So this piece of messenger RNA is about to leave the nucleus, the control center, and it's going to go out into the cell and start making protein. So let's take a look at what's coming next. Transcription, DNA is converted to RNA. The next topic is translation, and that's where RNA goes to protein. To do this step, you have to have your pink sheet, your codon wheel, or your codon table. This is where RNA goes to protein, and these are the steps. First, a ribosome, which is a little protein factory, binds to that template strand of messenger RNA that we just made. And then tRNA brings amino acids from the cytoplasm to that mRNA template and starts the process of translation. All this is happening in the cytoplasm. I'm going to open a link, or try to, there we go, for some animations that will show us transcription and translation. I'll just need to scroll down and find it. Give me just 1 second. Okay. This will load shortly. And I'm going to show you transcription and translation if this animation will work with me. That is. Okay. So here we have our DNA, and we're about to transcribe that DNA into RNA. That requires the help of the enzyme polymerase. Polymerase binds to it, unzips the DNA, and then bases get added. I don't know how well you can see this on your screen, but these are labeled T, C, A, C, A, C, etc., etc. So to be able to transcribe, I would have to match the appropriate base from RNA with the base on the DNA. So where I see a T on the RNA, the green one on the very far right, I'm going to add an A. If if it'll cooperate. There we go. So it added that one. And C will bind with G, and A will bind with U, and C will bind with G, A with U, C will bind with G, and then these 3 T's in a row all bind with A's. Now I'm adding in the bases. Watch what's happening to this chain right here. This is the RNA that's being made from the DNA. It's growing longer and longer, and it's kind of expanding out and will eventually turn loose and be released to the cytoplasm. Okay, so I'm going to finish this up. G will bond with C, A will bond with U, G will bond with C, A will bond with U, C will bond with G, and again, A will bond with U, T will bond with A, C will bond with G, 2 A's in a row, they both bond with U's. Sorry, this is a little tedious. G bonds with C, C bonds with G, A bonds with U, and T bonds with A. And by the way, if you would like to work with this animation yourself for practice, I can send you that link. Just let me know. So my DNA has been used to make a template, or used as a template to make mRNA. This mRNA is about to be cut loose. So let go. And what's going to happen to it? Will it leave the nucleus or will it stay there? DNA is too big to leave the nucleus, but mRNA can leave the nucleus. So we'll make that happen. RNA leaves the nucleus and it goes out into the cytoplasm, where it's going to be used to make protein. Now, these little dots, the orange, the blues, the reds, those represent amino acids. These little carriers here are transfer RNA that help transfer the amino acid into place. So it's kind of a matching game there too. In translation, a ribosome, which is 2 parts, will attach to the mRNA. And then transfer RNA, tRNA, will bring in the anticodon to help link the amino acids. We have AUG, which is the start codon. The anticodon for AUG is in one of these molecules. Anticodons are the opposites of the codon. So A would be U, U would be A, G would be C. We need to find something that has the anticodon U, A, C. I'll give you a second to look and see if you can find it on your screen. And hopefully you picked this one. UAC is the transfer RNA that will bind right here. And when it binds, it puts this little amino acid in place. The next little section gets read. CUU is the codon. The anticodon would be GAA. That corresponds to this one. And so this amino acid is brought in by this tRNA, and the 2 amino acids got joined together. Notice I have a little chain of protein being made, all getting joined together. I'm going to do one more, and then we'll be done with this and go back to our slideshow. GAU, the anticodon would be CUA. This one. When it's put into place, the ribosome joins the amino acids together, and eventually we would have this long chain of protein that would be released, and the protein would go out into the cytoplasm and be packaged up by a Golgi apparatus to be used somewhere else in the cell. So translation, I'll go back full screen now. Translation is copying RNA into protein. Let's see if we can practice this one. I want you to try to translate this. If you got a sheet of scrap paper or something close by and you want to do it there, that's fine. I'll be doing it on the screen for you. Remember, the only way to translate is to use RNA and to use your codon wheel or your codon table. The wheel was just the shortest one that I could fit into this slide. So my first codon is AUG. We know that means start, so I went ahead and put that there. If the next codon is CCU, I would find C, C, U, and match it to the amino acid. CCU corresponds to proline. Then we have CCA. CCA, also proline. The next codon is GAC. Find it on the wheel. GAC, aspartic acid. That's ASP. And I think you probably already know this one. UGA is the codon that means stop. So I'm going to type these in on the screen in the order that they would have shown up. Start, proline, aspartic acid, stop. And these three things join together as one protein. So translation. Proline, proline, aspartic acid, stop. These three would be released as a protein molecule that would go into the cytoplasm, get packaged up, and be used somewhere else in the cell, or maybe even outside of the cell. That brings me to a good point that I don't think I put into the webinar. What is the importance of protein? Protein is used to build new cells and new tissues. Protein is used in cell transport. It acts like the little channels through which material can move, and it acts as a support system. I gave you the example, like if the block walls in the classroom represent the cell membrane, the steel bars that are found in the wall for reinforcement, those are like the proteins helping hold the walls up. The doors in the classroom represent like protein channels to let things in and out. So protein is very important for all living things. And the way that we make protein is translation. Moving on. The other aspect involved in translation is tRNA. Remember, tRNA is the molecule that brings the amino acids into place. It does that using anticodons. So we need to be able to figure out what the anticodons are too. If this is my messenger RNA, the tRNA will be the opposite. The anticodons, in other words, will be the opposite of these. See if you can figure these out. The anticodon for AUG would be U, A, C. What about CCU? What would the anticodon be? Well, C binds with G, G again, U binds with A. So GGA. And then UCA, the anticodon, the tRNA anticodon would be AGU. If you have questions on the anticodons, just remember that anticodons are the opposites of the mRNA. They're both RNA, so we don't use T's in anticodons at all, because T's are found in DNA. But it would be the opposite of what's on the mRNA. Okay, kind of wrapping things up, I think the last thing to talk about was mutations. Before I move forward, you will need to know this statement on your screen right now. This statement for your extra credit. Two favorite colors, red and blue. One more time, two favorite colors, red and blue. You'll need that for the extra credit assignment. So let's talk about mutations. If this sequence on the top of my screen is my original DNA, I can have three different kinds of mutations possible. I can have a substitution, which is where one letter, one base, gets substituted for something else. The location of this substitution is this A right here. This A got swapped out for a G. So with a substitution mutation, one base gets swapped out for another. And it causes some problems sometimes. It would cause a problem if this GGG made a different protein than GAG, because then the cell would be affected. Sometimes it's called a silent substitution, because maybe GAG and GGG both code for the same protein or same amino acid. So the protein would be exactly the same and there would be no effect. We call those silent mutations. The second was an example of an insertion. We go AUG, CTC, just like in the template. But then there's an extra C added in that throws everything off. Let me show you a way to figure out whether this is insertion, deletion, or substitution. Or you can divide things into codes right there. And when you look at your mutated DNA sequence, you'll know it's an insertion if there's one extra letter floating around on the outside. An insertion happened. This C was put in. Didn't replace anything. It just got put in as an extra. That's an insertion. And then this one is a deletion. If you look at the anticodons, you'll see why. I'm sorry, the codon sequence, you'll see why. When we divide it up, it's divided into three-letter pairs, but one pair is missing one letter. So we know a deletion took place. What's been deleted? See if you can figure that out. Check the original against the mutation. AUG and AUG, that's fine. CTC and CTC, that's fine. AAA, AAG, that's the wrong section. So one of these A's got deleted and shifted everything to the left. So deletions, insertions, and substitutions are all examples of mutations. And again, some mutations are problematic, some aren't. So let's see if you can come up with the mutated sequences here. On the DNA, I have AUG, CTC, AAA, GAG, TAC. This is my regular old DNA. Let's see if we can figure out if this mutation is going to be silent. So my mRNA for this will be U. Oop. Did you notice this mistake? This DNA should not include U's. So I'm going to sub it for a T. Sorry about that. So the mRNA is going to be U, A, C, G, A, G, U, 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 C, U, C, A, U, G. Now, if I were to translate this into amino acids, I'd have to use my codon wheel, my pink sheet, and we'll kind of skip over that for right now because I think most of us are okay with that. But remember, you would find U, A, and C, match them up on the codon sheet or on the wheel, and put in that amino acid. Would it really start reading right here? No, because it should have started with AUG for it to start reading. But assuming that it did read, it would produce amino acids from these things. Okay, what would the mRNA be down here? U, A, C, G, A, G, U, 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 GGG would become CCC, and then TAC would become AUG. Because what's going on here is transcription, DNA being converted into RNA. I think we would agree, even if we don't look up the amino acids, I think we would probably all agree that everything's going to be the same, except for where this CUC got converted to CCC. This is going to be one amino acid, and this one would be something completely different, most likely. So this would probably cause an effect in the cell, and this would not be a silent mutation. So it would run into problems. Now I'm going to flip over to my question section because I'm getting ready to wrap things up. I'm going to flip over to the question section, see if you have any questions for me, and I'll try to take those questions now. Okay, so Christian, you said, how many amino acids are there? And Daniel, you've said, can you go over mutations? Hopefully what we've talked about with mutations is good enough to kind of cover your questions. Just remember there are three types. Christian, I'm coming to your question in just a second. There are three types of mutations: substitution, which is where one letter gets swapped out; insertion, where an extra letter gets added; and deletion, something got taken away. And then again, if we were to convert these into proteins, we would probably see that this amino acid right here and this amino acid right here, they're different. So that mutation causes a different protein to be made. And if the protein is different, it's going to function differently. And if it's functioning differently, then the cell will work differently. So mutations can be non-harmful. They could be harmful, as in they cause problems. And some mutations can even be beneficial. Sometimes things may mutate for the good. And we'll talk more about mutations and things in the second half of our genetics unit when we start talking about crossing things and breeding things. But hopefully those slides that we went through, Daniel, they will answer your questions about mutations. If not, I'll be available right before school and I can answer your questions there too. So just let me know. Christian, you asked how many amino acids are there? There are 20 different amino acids, essential amino acids in the human body. And remember that out of all those different amino acids, that they're the combination of three-letter codons. And since there are all these different possibilities, it could be GGG or GAG or GAC, all these different combinations come up to 64 different possible codon combinations. So there are 64 codons, but 20 different possible amino acids. Okay, I'm gonna get ready to kind of wrap things up here in just a second. And I'm gonna give you, since there's probably a little bit of a delay, I'm gonna give you about 1 more minute to submit any last minute questions. And even after the webinar closes, if you have questions, you can still key them into that box, because that's still gonna be live. You can still key your questions into the box on the online classroom right here. And I'll still get them. And if I don't answer them in the webinar, then I will have an answer for you when you come into class tomorrow. How about that? All right, I'll check back on that in just a second, see if any more questions come through last minute. But I want to remind you to go ahead, as we're wrapping up this study session, you can go ahead and get ready to close things down. I want to remind you to go ahead and complete the extra credit. If you've watched this, or if you're watching it after the fact, because this video will be put up on the online classroom even after the webinar is over, complete the extra credit. Click on this link and answer the questions in the extra credit form, and you should be good to go. And you have to answer the 3 random questions: the scientist, the animal that we talked about, that 3-letter word that kept being repeated in the replication sequence, and my favorite colors. You have to answer those to get credit. And then there's some actual content questions for you to answer. And assuming that you do all that correctly, you can possibly have up to 10 points on your test added tomorrow. One final thing that I'm going to give you is that your study guide for your key is posted here. Now, the key that is posted is slightly different than your study guide. So be sure that you look not just at the question number, but you read the question, be sure that it matches up appropriately. If you have any final questions, feel free to let me know. Come by and see me in the morning. I want you to do well on this test. So we'll review a little bit in the morning before we test. But if you have any issues that you want to talk about one-on-one, then I'll be at school sometime at least by 7:30. And you're free to come down and see me if you'd like to. Any final questions? How can mutations be harmful? Daniel has a question. How can mutations be harmful? If the codons, I'll go back to this screen. If the mutation caused a codon to be put in for stop, like if these 3 letters right here got converted, and instead of CCU, if they got converted to UAA, you look at the codon wheel, you'll see that UAA is a stop codon. So that one mutation would cause the protein to be start and then immediately stop. In other words, the protein wouldn't even be made. So if a protein's not made, there's something missing in that cell. It could have been the protein that makes the protein channels. So the cell couldn't get things in or out of the cell, and the cell would die. Or it could be that the protein that was not created was the protein that gives the cell structure, like in the cell membrane. So the cell might collapse inward. So they can be harmful for lots of different reasons. And I know that's really probably not a good answer to your question, but we're going to study more about mutations and how and why they're harmful, or how and why they're beneficial, in the second half of our unit after Christmas break. So I hope that was a decent answer for your question. I'm going to flip back and see if anybody else has submitted anything. All right. I'm not seeing anything else so far, but I'll keep a check on this tonight. And if you have any other questions that come through, I'll be sure that I answer them for you in class tomorrow. Okay. All right. Going back to this page, remember to fill in your extra credit, and that will come automatically to me. I'll check on that tomorrow when I start to grade the test. At some point over the weekend, I'll be sure you get the points for completing the extra credit. For those of you who didn't get to watch the webinar and you're watching this later, thank you for logging in and watching this after the fact. I know you have a busy day. Everybody's busy with sports and whatnot. So thank you, all of you who are watching, for taking time out of your day to try something out like this with me. I appreciate your help. And if this goes well and you've enjoyed it, then we'll try to do this again in the future. I hope y'all have a good night.